and you also will get that notice pop up. Sorry, we're plagued with technical issues this morning. Okay, so welcome everybody. I'm Susan Sang. I'm one of the co-PIs on the GBATnet NSF grant, and I am your host today for this talk, which will be delivered by our GBATnet student rep, Gwenddolen Kettenburg, who is a PhD student at the University of Chicago. And so I'm going to remind everybody again that this is part of the My Field for Dummies webinar series from GBATnet. So the main aim is to help people who are not experts in disease ecology understand the field better so that they can be, helpful collaborators in the future for you. And so I encourage people to share the link of the video after with their colleagues who might find it helpful, and just let people know. We will also post a talk onto the website under the resources pages, much like we did last week for the microbiomes, but last month for the microbiomes one. And they'll just all be listed on there, and you can access it however you want. And it'll be on the GBATnet channel, and I'll make a playlist for it and everything also later on. So yeah, today we are talking about disease ecology. This is a field that obviously has a lot of misunderstanding in some cases. A lot of people are not familiar with it, especially in the bat community sometimes when people are working from very different starting points. And so Gwen is going to walk us through that today. And I'm going to hand it over to her and also turn off my camera. Cool. Gwen, if you would like to start, feel free to do so.
Yep. Okay, cool. Thank you, Susan. So like Susan mentioned, my name is Gwenddolen Kettenburg. I'm a PhD student at the University of Chicago in Dr. Kara Brooks' lab. And this is the second installment in the My Field for Dummies seminar series. So today I'm going to talk about disease ecology, and I'll just get right into it. So I wanted to start off with kind of my path to disease ecology and how I got to where I am researching what I'm doing now. So starting off with undergrad, I went to a super small liberal arts college in a super rural area with lots of land around it in Pennsylvania. And I did intro research projects on bacteriophage resistance to E. coli, and a bacteriophage is a virus that only infects bacteria. And as a research assistant on the side, I did work with collecting ticks on campus and doing PCR for surveillance purposes for things like the causative agent of Lyme disease, which is a bacteria called Borrelia. And then in my microbiology class, I read a book called The Hot Zone, which is a nonfiction book, but it's very sensationalized about filovirus outbreaks such as Ebola viruses and Marburg viruses. And then I read that and I thought, okay, that's what I want to do. I want to go into high containment work, and I want to look for programs that did that. So for my master's, I picked a lab that worked on H5N1 avian influenza, which is a BSL-3 pathogen. So can still do substantial harm to humans, but not as crazy as something like Ebola. But I got to learn a wide variety of things, molecular tools. I did a lot of microscopy, which you can see here's a picture I took of a human lung infected with influenza. And then I worked as a lab manager in this lab as well to help pay off some of the master's degree costs. And I liked the work a lot, but I felt like I was not appreciating the pathogens I was working with for how they actually are in nature. They're not just always sitting in a tube in a lab. And so I wanted to go into more of an environmental focus. And so when I was applying for PhD programs, I wanted to work in a lab that did field, computational, and molecular work. So I was looking for anything having to do with disease ecology, and I wasn't necessarily targeting bats, but I'm extremely grateful I ended up working with bats. But for now, I finally feel like I'm in the research niche I was meant to be in. I love doing disease ecology work, and in this lab, I study how ecological factors contribute to viral maintenance. So how the virus just lives in this population normally, and what ecological factors can stress the host out, in this case, these 3 endemic Malagasy fruit bats, and increase the amount of viral shedding or the pathogen leaving the organism into the environment. So I'm fairly new to disease ecology, and so I feel like it's a misunderstood but very accessible field once you get into the terminology of it. So as a quick outline of today, generally I'm going to talk about what ecology is. I think that's a really important precursor to understanding disease ecology because it is a subdiscipline of ecology. And then I'll talk about disease ecology and what we do with that. And then finally, since this is a GBATnet seminar and this group all does research on bats, I'm going to talk about how disease ecology is applied to bat research and how there are very common miscommunications and stigmas that go along with that, especially in the wake of the COVID-19 pandemic. So starting off, generally ecology is the study of interactions of organisms with each other and their environments. For example, on the left here, there was a study of these 5 species of warblers that all share the same breeding grounds in the same trees. It was thought that all 5 species occupied the same space and competed for the same resources, but by observing their interactions with each other, we now know that each warbler species uses a specific part of the tree to forage for insects to avoid competition with the others. The field uses models to formalize general laws and principles that describe what they're seeing. So if we have an observation that lynxes prey on hares, we would call this a predator-prey interaction. And how can we use this observation to show what happens to each population over time? So we record the instances of lynxes preying on hares as data and then build a model from that, which is the basis of a predator-prey model. Both of these examples show that the study of ecology is really interested in interactions at its core and how we can describe those interactions using models. There are 2 types of models. There's statistical models, which are pattern-driven, and there are mechanistic models, which are process-driven. Statistical models are like I said, pattern-driven, but also data-driven with the goal of finding patterns of correlations. And then mechanistic models are process-driven with the goal of understanding what happened, when it happened, and why it happened. So for example, here on the left is a graph of forest cover over time. And so we can see that there appears to be a pattern of forest cover decreasing over the years. Whereas on the right, we're trying to explain why this might be happening. So you have a forest and it has some rate of growth that allows it to keep growing as a forest, but then you have some other rate that's causing it to decline, such as slash and burn is what we're hypothesizing here, which is causing the forest to turn into a savanna type of habitat. So we can transform the observations we see in the environment, which can tell us something about the patterns and processes in ecology. But nature is random, so of course our collected data isn't going to form a perfect straight line. So we need to do something called model fitting. And so, for example, to make a model, to start off with, we still define a research question, formulate a hypothesis, and collect the data. And then we construct the model that would demonstrate our hypothesis. So when we do the model fitting, we're testing these different hypotheses and asking, assuming our model is true, how likely are we to recover the observed data? And we do this by optimizing the parameters behind the model and get it to fit to the data. So all the way on the left here, this example of the tree, the forest cover over time again, we have a couple of different lines, and these are different models we're testing. So the pink line all the way at the bottom doesn't seem to match up super well to our data. So we would say that's not the best model. That's not matching our hypotheses. Whereas the blue and looks like black lines above and below the data are certainly closer and kind of represent the direction of the data, but they don't match super close to the data. So they're also not the best model. Whereas this reddish brownish line that goes directly through the data best encompasses what we're seeing in the data. So we would say that's the best model. So once we have the best model, we can use this to explain and predict what might happen with our data. So this middle example here, With a model, we can explain how prevalence in some sort of pathogen here, I think this is from a paper on coronaviruses, is changing over 1 calendar year. So this is looking at the seasonality and prevalence of subtype of pathogens. So we can explain what might be happening there. And then using these models, we can predict what might happen in the future. So many climate models forecast what will happen after a certain amount of years, using data that we've collected in the past and up to this day. And disease modelers also sometimes use forecasting models to predict what might happen. So early on the COVID-19 pandemic, people tried to use current and previous case data, although limited, to try to predict what would happen next with the pandemic. And ecology can be studied on a range of different scales or levels, and you can think of these as units of measurement. We're interested in these levels can range from something small, such as one certain individual from a certain population, all the way up to the biosphere, which is just the planet. And to get into these individually, on the individual level, we can study adaptations or features arising by natural selection that indicate the organism is changing with their environment or some other factor. And we can, for an example, we can ask, how would this individual Mexican free-tailed bat adapt to climate change? How would this bat compete with another bat for food? And when I talk about adaptations, there's this really famous example of these moths. I don't remember exactly where, but in Europe, they were one certain color. And then I think there was more smoke in the environment at some point, and the moths were able to be seen by birds. And so eventually the moths adapted to becoming a darker color in the darker environment to avoid predation by birds. So that's an example of adaptation. The next level is a population, which is a group of organisms or individuals of the same species that live in the same area at the same time. Population ecologists study the size, density, and structure of populations and how those change over time. So, for example, this is a colony of Mexican free-tailed bats. And we can use these this metric to these population numbers to determine, if a population is stable or changing. And another example would be to think of humans. Our population is growing every year. So again, just one, a group of organisms of the same species at one point in time. And so another higher level is a community level. So a biological community consists of all the populations. So we had the individual bat, we had the bat colony, and now we have these populations of these different organisms that live in the same given area. And community ecologists focus on interactions between populations and how these interactions shape the community. So as an example of what kind of question we can ask, when there are more moths for bats to eat, does the bat population increase? And you can think back to the predator-prey example with lynxes and hares. How does the amount of lynxes around, which are the predator, affect the amount of hares, which are the prey? And in the predator-prey models, you can actually see that when there are a lot of lynxes around, the hare population decreases, but then when the hare population decreases, there's not enough food for the lynxes, so their population decreases. And it's just a staggered, like, up and down of the population over time of these two different populations interacting with each other. On an ecosystem level, it consists of all the organisms in the area, the community, and the abiotic factors that influence the community. So an abiotic factor is something that is not alive. So you can think of this as weather, soil composition, temperature, stuff like that. And so ecosystem ecologists often focus on the flow of energy and recycling of nutrients in the system of study. And an example of a question we could ask is, how does rainfall affect the populations of bats and moths around Bracken Cave? And then finally, at the biosphere level, the biosphere is a planet Earth, viewed as an ecological system, and ecologists working at the biosphere level may study global patterns, for example, climate or species distribution, interactions among ecosystems, and phenomena that affect the entire globe. And the best example is climate change research, because it affects the entire planet and all the populations and communities on the planet. So with all that information, I'm hoping that I know ecology can be used to answer many different questions, but again, mostly focused on interactions of these organisms in their environment or with the environment. In bat research, ecology is used to determine many different things. For example, their important ecosystem roles in insect predation, seed dispersal, and pollination. There are also longitudinal studies that can determine seasonal variation in bat size and health, which is the plot all the way on the right. Both at the individual and the population level, and these are for 3 different species for males and females. And then you can also do population viability analyses, or a PVA, which are used to understand extinction risk and forecast future scenarios of population growth and decline. There's another term called minimum viable population, which is another metric to determine the smallest population size that will persist for some specified length of time. And these measurements are also used in conservation biology. For example, PVAs are used to inform decisions on what bats are classified as on the IUCN Red List. For example, one of the bats that we study in Madagascar, it's considered threatened, I think. And then in doing a population viability analysis, we would doing consistently population viability analyses, we'll be able to look at that metric over time and see how the population is changing. So with all this information, we can get into, so what is disease ecology? Disease ecology is a subdiscipline of ecology that draws inspiration from many other disciplines. It's still a form of ecology, so the goals of disease ecology align more with conservation research and population and community ecology more than one may think. Both conservation biology and population community ecology want to protect species. That's the main goal of doing the science of both of these, but also studying the interactions of organisms with each other. Disease ecology draws from other outside disciplines, like I said, to help interpret the data. One thing it sometimes pulls from is public health, which is a field that's concerned with protecting the health of people, humans only really, which is a human-centric view, but comes into play when we think of something that occasionally infects humans, like avian influenza or an Ebola outbreak. And One Health is a fairly recent focus. I think it was only defined in the past 30, 40 years. It was something pretty recent, but it calls for collaborative efforts of multiple disciplines working locally, nationally, and globally to attain optimal health for people, animals, and our environment. So trying to link all these fields together so we can all kind of work together to work towards a common goal of protecting the health of many different organisms and species. But disease ecology focuses mainly on the environment, which is the main definition of it, and wildlife and how hosts, which are animals, interact with their microbes or parasites, which are these here. And molecular biology is another thing we use, but that's kind of what I said I was doing when I was working with influenza. I was only looking at how this pathogen, like what factors of it could influence something else, not in a larger scale of the environment. So all this information together can help us understand patterns and processes we see with diseases. So one of the main questions we ask is, how do climate, evolution, and free living hosts interact to determine the dynamics of pathogens and the burden of disease? And it all comes back to interactions here. Disease ecology strives to understand the mechanisms and the scale of pathogen impacts on host individuals, populations, communities, and ultimately ecosystem function through the multidisciplinary approaches I mentioned before. And some examples of disease ecology are when people describe things like amphibian chytrid fungus. So this is a devastating fungus that is wiping out a large amount of amphibian populations and is reducing biodiversity. So disease ecology has found that the temperature of the water around the area can impact how well the fungus grows. And so by looking at these interactions, we can determine possible interventions to how to protect the amphibian population. Lyme disease is another well-described pathogen in disease ecology. It's spread both to animals and people via bites from ticks. And so we're focused on just how the tick population impacts mammal populations and human populations. And then on the right, parasites that just live naturally within these sheep keep the population in check on an island. So on certain years, the population will like crash dramatically and then be able to recover. So pathogens, microbes, I'll define those in a minute, all play an important role in population regulation as well. But the main point of confusion is what is meant by disease? What is a pathogen? Are we all talking about the same thing? And unfortunately, it can mean many different things depending on the context and of what is being studied. But here I'm going to define some of the terms to hopefully clear up some of that confusion. So there's this great paper that went out by many members of the GBATnet community in response to the COVID-19 pandemic, which fostered inaccurate communication from scientists, leading to people thinking, at least the COVID-19 pandemic led to people thinking all bats everywhere are bad. And so this is a great piece of literature which defines terms, how they're currently used, some miscommunications, classes of miscommunications that these terms can be, and how we can use them in the correct way. So I'm using a lot of terms from this paper. Some terms from it may not come up in this talk today, but it's a very informative read if you want to take a dive into this later to understand a lot of the disease ecology terminology and where a lot of the miscommunication with the field comes from. But starting at the smallest level, like we were talking about with ecology before, there are different levels of terminology in disease ecology. So I'm going to first describe the organisms that we study. So there are, here are the common types of microbes, which are defined as not shown to cause damage or disease to the host, which could become pathogens, which are shown to cause damage or disease to the host. So these are the common types of microbes. I'm not sure if you can see my mouse, but they're grouped by cellular living or acellular non-living. Many of these, almost all these, are parasites because they require a host to complete their replication and life cycles. But microbes is a very broad term that's usually referring to any of these organisms listed below. But like I mentioned before, in an effort to clarify these terms for general use, as pathogen has negative implications about it. If someone hears that something's a pathogen, they think, oh, that's going to hurt me. And it may not hurt you. But as an example, it's suggested to use microbe when it's not known to cause damage. So using an example from here, I listed some of the common examples of these different microbes. So for example, the protozoa that results in malaria in humans does not cause damage to the mosquito it lives in. So we would only refer to it as a pathogen in reference to humans. So as we said before, pathogens should be used when referring to the damage the microbe caused to the host. So using that example again, malaria is a pathogen to humans. It's a microbe to mosquitoes. And part of the big issue with the field is that scientists need to be careful about defining a species as a host or a reservoir for a microbe. And I'm going to get into those terms next. And so for the next level up of what these microbes cause or what they live in, we mentioned that most of them aren't free living. They need a host to replicate and survive. So as I just mentioned, a host is an organism that houses the microbes and allows a place for them to replicate and live. Disease itself is defined as a pathogenic condition of a host, sometimes caused by a pathogen or parasite, and thus diseases are not transmitted between hosts, but pathogens and parasites that cause diseases are. So I'm going to use COVID-19 again, just because I think it's an accessible example that many people are very familiar with. COVID-19 is the disease in humans that is caused by the virus SARS-CoV-2. So COVID-19 cannot spread between people, but SARS-CoV-2, the virus that causes COVID-19, can. So disease only refers to the condition in the host. But reservoir is defined as a population, species, or community in which the microorganism naturally occurs and is indefinitely maintained. This is one of the terms that is abused a bit when talking about disease ecology. And the Shapiro paper mentions that this is one we particularly need to be careful about, as it's stigmatized bats especially. But calling something a reservoir species involves a ton of work and a ton of field work, molecular work that may take a long time to determine. And so just calling something a reservoir species because you think it comes from there may not be correct. And then vector is a specific type of host. Vectors refer to arthropod hosts and microbes like ticks, flies, lice, stuff like that. And then shedding is the expulsion of disease-causing microorganisms into the environment. And so I mentioned before that a lot of my research focuses on shedding and how the environmental factors cause the reservoir host or host, has not been reservoir host or host, depending on the pathogen, how environmental factors mediate this amount of shedding. And then, so hopefully with all these defined, we can see that disease ecology is interested in microbes that do or potentially have the cause to harm hosts, and how environmental factors affect the spread from one host to another. So now I'm going to define some of the processes in disease ecology. Spillover is one of the first terms, and this happens when a microbe is transmitted from a reservoir host to a spillover host, either directly or via a vector. And it happens when a pathogen transmits from one species to another. It could cause harm to the spillover host, or it could do nothing. The term spillover refers to one single event where, and these are very, very rare events, the spillover host is usually a dead end host, meaning that it will accidentally spread from the reservoir host to the spillover host, but usually will not persist. So it's almost impossible to quantify the chance of this happening. I tried really hard to find an actual number to give you, but I couldn't. But it's super, super rare because the microbe needs certain factors to all align for the ideal environment, and there are super significant barriers that it needs to overcome. So it depends on certain factors can increase or decrease the chance of this happening. So it depends on the spillover host behavior, how, in this example, humans, how are certain factors allowing them to interact more with the reservoir host, which may mediate the spillover event from happening. So an example is avian flu. Farmers that come close with poultry have a chance of getting infected with it. To date, it is not spread from human to human, and these spillovers from the poultry into a human are rare, but farmers who have poultry and interact with them on a daily basis have a higher chance than the general public of getting infected, but both of those instances are still low. And for example, as we mentioned before, disease ecology cares about environmental impacts and how the microbe spreads. So things such as deforestation could remove natural habitats of reservoir hosts and allow for more interactions with a spillover host, such as a human, for example. And then there's a term called zoonoses, which refers to a pathogen that's transmitted from an animal reservoir to a host. So over 60% of all emerging infectious diseases are zoonotic, meaning transmitted from an animal reservoir to a human host. And this can happen either directly or in this example, which is kind of showing plague, can happen via vector, where the normal reservoir host indefinitely maintains the microbe. Something like a vector, like a flea, bites the reservoir host, and it then is transported to whoever the flea, potentially transmitted to whoever the flea bites. And the same thing happens with tick-borne diseases as well. But while many emerging infectious diseases are zoonotic, you still have to remember that spillover events need to happen and the pathogen needs to be able to persist in a new host for it to be able to take off into anything similar to an outbreak, an epidemic, or a pandemic where there's sustained spread in the new spillover host. So this is a spillover, but it's specifically referring to transmitted from an animal reservoir to a human host. But not all spillovers are zoonoses. Spillover can also happen in alternative wildlife hosts. So rabies is a good example of this, as it infects a large variety of animal hosts and can transmit to other animal hosts. So domestic dogs may carry it and transmit it to a wild animal and may allow a sustained spread later on. And then there's this less commonly known term called spillback. Sometimes I've seen it referred to as reverse zoonoses. And this is where pathogens transmit from the spillover host back to the original reservoir host, or in most of the time it's from an animal back to a human. So an example of this would be two instances that happened during the COVID-19 pandemic. There were minks in Denmark or the deer in the United States that were found to be naturally infected by SARS-CoV-2 from humans getting close to them as the virus spread globally. But this is also an interesting example because humans are not the original reservoir hosts of SARS-CoV-2. We still don't know what that is, but it's an interesting example nonetheless. It's mostly a less known term because most of the time, the general public and people think of pathogens spilling over from animals to humans and not the other way around. And then spillback occurs among wildlife as well. It's not just with humans. For example, cattle sourced Brucella to wild ungulates in Yellowstone National Park, which now serve as the source of reinfection to cattle. So these were the original reservoir hosts. They introduced it to wild hosts, and now they maintain it in their population and can spread it back to the original host. So now we know, or at least have gone over a lot of the terminology used in disease ecology, but we mentioned before that ecology uses models to show, demonstrate what's happening, demonstrate observations and predict patterns in data. So how do we do that with disease ecology? There's a certain type of model called susceptible, infected, recovered, or SIR. And these put the study organism into classes and shows how they transition from one class to another at certain rates. And so here you can see the proportion of, let's say, people infected over the course of an outbreak. And so you have the amount of susceptible people starts off at 100, but then it drops as the amount of infected people goes up. And then as people clear the infection, they recover. So we still fit models to data. We still use both statistical and mechanistic models to make hypotheses about how the pathogen or microbe will persist within a population and spread. And we still do model fitting to determine what's going on in our data. So in this example here, various versions of the SIR model have been proposed. So these are our hypotheses. M refers to maternally immune. So you can modify your SIR, susceptible, infected, recovered model to better encompass naturally what happens over the course of disease in your study population. But these various versions of SIR models were proposed to describe how henipaviruses are maintained and spread in populations of this Malagasy fruitbat, E. eidolon duprinum. And using these models, we were able to determine the importance of maternal immunity being conferred to bat pups. Now we know mechanistically how a pathogen behaves in this specific population. We can make predictions about when shedding might be highest, if spillover is likely to happen, if people are even getting close to these species, or when susceptible members of the population are at most risk of becoming infected, which in this case would be bat pups, as they have not been exposed to this specific virus before. And then what kind of data can we use in disease ecology? Just as was shown earlier when discussing ecology generally, the level of which we're studying determines what kind of data we get and what inferences we can make. So on a population level, here's an example where you can say, host populations that were healthy from one year to the next were significantly smaller population sizes than host populations in which disease was constantly present. So by studying disease dynamics, we can possibly make inferences about the size of the population. On an individual level, we can determine, very generally how this is affecting that individual. So here we can see what is the viral load, or how much that virus is replicating in days since system onset. And so, this studying on an individual is not inherently disease ecology. Individual level data can be aggregated or combined together to determine population trends. And then finally, on a microbe level, we can use data such as how the pathogen is evolving in its reservoir host and how that impacts the health of the population. There's this classic study on the evolution of virulence, or the ability of a pathogen to cause damage to a host, with something called myxomatosis virus, which was originally released in wild rabbits as a population control in the 1950s. Rabbits that survived the initial wave of disease developed resistance, and in turn, the virus also evolved to overcome the rabbit's resistant mechanisms. So on a microbe level, we can show this interplay of interactions between the host and the pathogen as well. So looking at interactions between the host in the environment, the pathogen in the environment, and the hosts with the pathogen or microbe, we can determine the dynamics of how disease is maintained and spread within a population. We can also use disease ecology to protect the health of populations. One example that is pretty specific to tick-borne diseases, Lyme disease, is something called the dilution effect, which implies that where species vary in susceptibility to infection of the pathogen, higher biodiversity or more mammals that can possibly be infected are around, there's actually less Lyme around. Whereas if there's only one type of host, less biodiversity, more disease or more pathogen is in the ticks. So because high host diversity dilutes the impact of the ticks biting the white-footed mice, which is their main target. And if there's less biodiversity around, there's more of that target species around that could possibly be infected with the pathogen. So we can hopefully implied or shown by now, the environment is directly tied to ecology. Human-induced environmental changes such as landscape fragmentation and predator suppression can inadvertently increase disease risk by reducing biodiversity, as seen in this example. We can use disease ecology to make decisions to protect the health of populations by tracking how disease persists in the population. We can forecast what will happen in the future and come up with intervention strategies such as the appropriate time to vaccinate dogs to prevent rabies from spreading to people, and determine what percentage of the population needs to be vaccinated to prevent outbreaks. So with all of this, now we're going to get into how disease ecology is applied to bat research. So in disease ecology, bats are quite interesting as reservoir hosts, or millions of years of evolution have allowed them to form really close evolutionary relationships with certain viruses. And it's been shown that certain factors of their immune systems are extremely different from other mammals that we see, and constant stressors such as flight also impact their immune system, and in turn how they host these microbes or pathogens. But I think there will be another My Field for Dummies seminar on immunology, so I won't talk too much about that, and I'll leave that discussion for then. But bats have also kind of gotten a bad rap for this type of stuff because some, not all, but some high-profile spillover events have been linked to bats. So, for example, Hendra virus, which is a hennepavirus in Australia, spills over from bats into horses if they're in close contact with each other. And then sometimes Marburg virus, which is a filovirus in the same family as Ebola virus, can sometimes spill over from bats into primates. And this has piqued interest as well. Humans in general have more interest in things that affect themselves. So these high-profile spillover cases that have high virulence, or how much harm it does to the host, cause unnecessary fear. But there's also a research bias into viruses as opposed to fungi and pathogens in bats. And there's also a larger geographic sampling bias. So there's a huge disproportionate amount of research into viruses. There's a huge sampling bias in African countries and Asian countries. And so, and another thing is that we don't know how many of these, if we're looking at viruses or any other pathogen, just as there's a distinction between our microbiome, which are microbes that are normally there, it is unknown how many of these viruses that are found in bats are part of their virome, or viruses that are normally there and not, they're meant to be there. But there's also been research suggesting that species-rich reservoir groups host more virus species, and therefore a large number of zoonotic species. So bats may not inherently have more what we consider pathogens. They just have a higher diversity of species, and therefore may just have coincidentally a large number of microbes or pathogens. So for the rest of this talk, I'm going to focus on work that's being done so far in disease ecology and where the disease is heading, and more specifically, benefits of disease ecology in the context of bat research. So I mentioned this paper before, which has been put out to dispel some of these issues with miscommunication. And unfortunately, both disease ecology and bat disease ecology have gotten a bad rap. Lots of groups are working to fight these stigmas and working to protect bats and the researchers that handle them. So, for example, lost my train of thought. Oh yeah, sorry. Bats play extremely important roles in ecosystem functioning. So while the gut reaction, if there's some high profile spiller event of many governments, is to remove what they perceive as the problem, or issues such as culling or killing bat populations, this harms both the bat population, the ecosystem, and as you can see in the paper on the right, this may even lead to higher chances of a spillover than when the population was left alone. Another source of stigma, which is touched on in the Shapiro paper, is miscommunication over the capacity of what data can tell us. So with Marburg virus, which is in the same family as Ebola virus, one particular species of bat has been definitively described as the reservoir host. That is the bat where this virus is normally circulates. However, for Ebola, many people speculate that the likely reservoir is also a bat due to it being in the same viral family and through something called antibody data. But antibodies only tell you that the organism was exposed to the pathogen. It's not enough to say that it's a reservoir. And so a lot of times through these inferences, bats can get a bad rap for being a reservoir for something that they're not. But on a more positive note, I hope that the rest of these examples I showed convince you that this type of research is important. And as many other areas of bat research, disease ecology wants to protect the habitats of bats and protect them from harm. So for example here, over 20 years of Hendra virus outbreak data. This data shows that bats are responding to environmental change by adopting behaviors that were previously not their default behaviors in response to nutritional stress. So interactions between land use change and climate now lead to persistent bat residency in agricultural areas where horses are. And so that could increase the risk of a spillover from a bat to a horse. And this happened when there were food shortages that drove the bats to agricultural areas where there may be more food. But they also found that pulses of winter flowering blooming prevented the bats from moving into agricultural habitats and thus lowered the risk of Hendra spillover from bats into horses. So we can see here that protecting their habitat and preventing, making sure they don't lose things that they depend on, such as their food sources, is important for both them and from a disease ecology perspective. And on the right, it's been shown that habitat disturbance can stress bats out and weaken their immune system. Other studies have shown that stressed bats, whether it be from nutritional stress or something else like the process of birth, can shed higher levels of virus. So while it may not inherently infect something, while it may not inherently be a spillover, these stressor events are generally increasing the amount of, in this case, virus, because those are the studies I could find on increasing the amount of viral shedding going on. And then other stressors can be important for studying disease ecology in bats. We mentioned the study on the left shows how mercury and other heavy metals in the environment affect the pathogen and the vector directly. This is not a viral disease, but the same kind of effect happens where the environment affects it, which in turn affect the bats that can be infected. And the study on the right found that generally mammal species that harbor more pathogens overall are more likely to occur in human-dominated ecosystems, and they conclude that global changes in the mode and the intensity of land use are creating expanding hazardous interfaces between people, livestock, and wildlife reservoirs of specifically zoonotic diseases. Remember, those that can go from an animal to a human sometimes. And then there are pathogens. So another misrepresentation is that bats don't ever get sick from pathogens that they have, but some pathogens do hurt bats. And one of the biggest examples is white nose syndrome, which is a fungal pathogen that's devastated United States and Canadian bat populations. And from studying the disease ecology, people have been able to determine intervention strategies, and they've tried things like adjusting the cave temperature to prevent the fungi from growing, or in this study, bats that aren't as nutritionally stressed and have higher fat stores going into hibernation have lower prevalence of white nose syndrome. And then other studies such as rabies can make bats sick too, but it was super hard to find those papers so I could find the exact amount. If you Google, does rabies cause illness in bats, everything on your first page is an article about how rabies can spread from a bat to a human. But there's some very old reports of bat die-offs which were found to be infected with rabies. And this one study linked here found that 12% of deceased European bats were thought to die off from a fatal infection of either a virus, a bacteria, or a parasitic infection. So while many people think that bats never get sick, they certainly do, and these also need to be studied to protect bat populations. And then also we can determine interventions we can use to protect the bats, protect the populations that they threaten. And so in some areas, especially in Latin America, vampire bats have gotten a bad rap for spreading rabies to cattle through blood feeding. An unfortunate intervention that was developed for this was targeting the behavior of bats rather than understanding the disease dynamics. So something called vampiricide was developed to take advantage of the fact that the bats groom each other. And so what they would do is capture a bat, spread this poisonous paste on it, and then when the bat returns to its colony, it spreads to the others and it kills the bats. But like the Marburg like people trying to prevent bats in the caves to prevent Marburg outbreaks, doing this sometimes increases the amount of rabies virus spillover that happens from the bats into cattle. So as an intervention, we could take advantage of the same behavioral thing we've noticed, this grooming mechanism, and develop a transmissible vaccine. So instead of culling the population, which may increase the proportion of bats that are infected with rabies, the spreadable vaccine would increase the amount of ones that end up being immune to it and therefore not possibly not be able to spread it to the cattle. And then finally, a lot of bat virus research uses surveillance combined with ecology. And this is a lot of what our lab is doing. Longitudinal studies are the key to making these long informed analyses on disease ecology of a pathogen. However, getting this data requires a huge amount of effort and time invested into collecting this data over the years. But one example of using surveillance in concert with ecology, we collect samples monthly, and we use demographic data collected at the time of capture to determine birth pulse timing for these 3 endemic fruit bat species in the paper I showed earlier, and these seasonal variations in mass to farm ratios. And we have antibody level data for certain viruses that we can correlate to these environmental factors. So, for example, we could use this data of when a certain pathogen, in this case viruses, which is what we're studying, is shedding the most, when a birth pulse is supposed to happen, we could possibly design a wildlife vaccine to determine the best time to administer it. And by doing this surveillance in concert with ecology, we can design better assays to pick up area-specific pathogens and more efficiently determine finer scales of variations in disease dynamics, and also contribute to surveillance for new pathogens or just seeing what's going on in the system. But temporal over time and spatial limitations in global surveillance and the lack of longitudinal studies is a main limiting factor in the type of data that disease ecology can show. I know I'm going a little over time for questions, but I'm finishing up now. But in summary, disease ecology encompasses aspects of population and conservation ecology. More work needs to be done to reduce the bias in disease ecology research, but especially bat disease ecology research. And limitations in the field are beginning to be addressed. Longitudinal, temporal, and spatial data can help predict potential spillovers and determine the cycling of the pathogens or microbes within these bat populations. And then finally, environmental variation in interactions impacts populations and their associated pathogens. It's just as important to disease research as focusing just on how the pathogen or microbe interacts with the host. So this time, I want to thank you for listening. And if you don't have any questions today, I'm more than happy to answer any questions you have via email. So thank you for listening.
Great, thanks, Gwen. If there are any questions, please raise your hand or type it in chat, whichever you feel more comfortable with, and I will call upon people if there are any questions. Is there a hand raised? No. So just to follow up with what Gwen was talking about in terms of their work, GBATnet also has one of their working groups that is related to disease ecology, and that you can join if you check our projects page where all the interdisciplinary projects are listed. So, fill out the form, and you'll get added to the members list, and the folks who are the working group leaders will get in contact with people whenever they're meeting and talking to everybody. And there's other groups that might be of interest to you if you have other interests. And so I encourage people to check that out on the GBATnet website. There's a question in chat from Caroline. What are you most excited about in your research right now?
Oh boy, a lot of things. I'm super interested right now about how certain host factors such as bats live a really long time for their age, and they undergo a certain amount of like cellular stress levels, but also with these close evolutionary relationships between bats and their virus. I study viruses only. I do like other pathogens, but since that's what I study, that's what I talk about the most. But I'm super excited now about determining how these stressors may explain why bat viruses may be virulent when they spill over into a non-bat host. But that's something I'm super excited about right now and something I'll work on in the future. But right now I'm also enjoying doing a lot of surveillance for these certain types of viruses to eventually correlate it to environmental data to determine when shedding is highest. So
Yeah, just a minor correction. When you said bats live very long for their age, I think you meant bats live very long for their size.
yeah, you're right. Like obviously they live very long if their age is long. Thank you, Susan.
Are there any other questions from anybody? All right, then. So as a reminder, this is going to go up on the GBATnet website. And next month, we will have our immunology talk, hopefully. If not, it will be at some point, there are multiple topics coming up, but I'm hoping the immunology one is ready by next month. And that will be delivered by our GBATnet student rep, Ty. And his background is mostly in things related to microbiology and immunology first, and he's entering into bats now. So he's going to be a really good resource for understanding immunology for everybody. So hopefully I will see you all there next month. And if you want to contact Gwen or contact any of us or leave a comment under the YouTube video, if you have questions, we will get back to you. And hopefully this was very informative to people. And please let us know if there are any other suggested topics that you would like us to cover in the webinar series. Thank you for coming.
Great job, Gwen.
Thank you, Teresa.
All right, I'm going to stop recording now.